I introduced to you John Vicente, who's a PhD postdoc at the Hawaii Institute of Marine Biology, and he's going to be, take it away, John.
Yeah, sounds good. I want to thank everyone for joining me today. The goal of this project is to establish an online database that has genetic resources and morphological resources for state managers to use for detecting the future arrival of invasive species through ballast water, and through eDNA analysis of ballast water, or even surveys through transects and field work. But before we begin, sponges get a bad rap here in Hawaii because the only thing we know about sponges in Hawaii is that a lot of them are invasive. When they come here, they haven't been found before, and the fact that they arrive here and take over the reefs allows us to see them in a negative context. However, they actually have a lot of very important ecological functions. They're very old metazoans, been around for more than 500 million years, and there's about 8,500 species that have been described worldwide. One of their most important ecological roles is the fact that they can pump very large amounts of seawater, and they can associate with a diversity of microorganisms that shape the composition of that seawater. So if you have 100 kilograms of sponge, you can turn over the ocean water in a coral reef environment of an Olympic-sized swimming pool in about a day. So they have really important ecological functions at the ecosystem level. And one big question which they have been contributing an answer to is is that question that Darwin was asking way back when, about how coral reefs can sustain high biodiversity in such nutrient-poor conditions. It's known as Darwin's paradox. So how can these ecosystems that have high biodiversity sustain this biodiversity, yet are so oligotrophic in nature? One answer to this is offered by a sponge. And this is a sponge that doesn't grow much. It's a sponge that grows probably the size of my hand. It's a fairly encrusting small sponge. And the reason why it's small, it lives in cryptic space, is very different from the sponge that I showed in the first slide. It has a giant, massive morphology. The reason why the sponge grows so small is because it is able to take up dissolved organic matter, which is not available to other organisms on the reef, fix it in the form of quanocyte cells, and it sheds these quanocyte cells into the marine environment in the form of sponge poop, also known as particulate organic matter. These detritivores are known to fuel higher trophic levels. So by fueling the lower trophic organisms, higher trophic organisms are able to feed on these and stimulate biodiversity that way. So sponges are very efficient at doing this. The red line on the left of this figure, the sponge loop, is basically dissolved organic carbon being transferred into POM. They do this 30 times faster than bacteria do it in pelagic systems. So they're very efficient at doing this. So how does this relate to coral reefs in Hawaii? Where we have coral reef dominated habitats rather than sponge dominated habitats as we observe in the Caribbean. Well, the reason is because if we were to flip this reef on its back, we would find this. And what we're seeing is basically more than 15 sponge species colonizing these cryptic communities of these coral reef environments. So the fact that we're not seeing sponges doesn't mean they're there. These are cryptic animals that live and fit that profile of this sponge that contributes to the POM loop, to the sponge loop. These are cryptic sponges that don't grow much, and there's a reason why they're not growing much. Potentially, they're contributing to this particular organic matter pool that's stimulating biodiversity. This is another way that sponges contribute to potential biodiversity, and it's consumption by predators like cowries, as observed in this video. These are cowries that are foraging on a Dicedia sponge, and the Dicedia sponge waste is actually contributing to the detritivore community, as observed by the detritivore in the background, which basically consumes the remnants of the cowries, which are just sponge organic matter. They also feed turtles, and we have found a very unusual observation in reefs in Mauna Loa Bay, where Hawaiian green turtles are actually observed not feeding only on algae, but also on boring sponges. And this is the boring sponge from Hawaii that we have found to be an important diet composition of the Hawaiian green sea turtles. Hawksbills also consume sponges, but this was a very rare sighting by Hawaiian green. And so they're a major food source for endangered species. Now, they have many benefits, provide many benefits for coral reef habitats. However, we know from previous work in other areas that they can arrive to areas where they're not from and actually take over, as has been observed with the sponge Terpio soshinota in other coral reefs around the Pacific and Indonesia, Japan, the Northern Marianas, where the sponge associates with photosynthetic organisms. It derives its carbon and food source from these organisms that they grow on and can actually wreak havoc on coral reef systems. What you're seeing is a brown mat just overtaking corals and basically killing them. And it's because of their association with these photosynthetic organisms they can grow so quickly. We also have boring sponges that can actually dissolve the calcium carbonate of corals, and this is actually in the islands of Chagos, where Cleona species are known to form these mats. They also associate with photosymbionts, but it's symbiodinium, it's cyanoflagellates rather than cyanobacteria, which can actually create these mats of bioerosion. And there's a second sponge at the bottom, there's Phonodictyum, that does the same thing. Then in Kaneohe, we haven't had any phototrophic sponges or any boring sponges that are invasive that are wreaking havoc, but we have these species that are palatable to predators, they lack chemical defenses, but grow very quickly. And once they arrive, they have the ability to displace corals as observed with Michaeli grandis. Although the invasive nature of this sponge is questionable nowadays because it's facing some reductions, but I'll get into that a bit later. So it's really important to understand where sponges are, where they came from, where they are around the world in order to actually understand where they're endemic to, where they're natural to, and where they come from to determine who are the invasives and who are the natives. However, we have a really big problem on a global scale since we really don't know what the diversity of sponges, of the global diversity of sponges actually is. So we can see by this chart, we can see that there's no way that you're going to tell me that the highest diversity of sponges is in the Northeast Atlantic and the Caribbean Ocean, and the numbers dwindle in the Pacific, especially in the coral reef triangle area. And in Hawaii, you see that it's you got about 60 species, and this is biased towards collection effort. So these areas in high red are red because people have dedicated a lot of work to the collection of sponges in these areas. So this brings a big problem, and some of the problems rely on very simple things such as taxonomy and taxonomists. We're losing sponge taxonomists because we're not funding taxonomists to do this basic work, and this has high consequences for fancy genomic tools like eDNA barcoding and metabarcoding. So, just to give you an example of how this might affect our advanced technological sequencing of eDNA analysis, this is by no means the details of what eDNA barcoding and code, but just to give you a rough sketch, we filter seawater. It has DNA from organisms all around the reef. We extract DNA from these filters. We use universal primers to amplify specific regions of DNA that will give you enough copy numbers to determine the organisms that are found. We sequence these products, and we align these sequences through bioinformatics with online databases that we hope are well curated and we hope are well regulated. And this gives us a species ID of the organisms that are found on the reef. The problem with sponges is that there's no such thing as a universal barcode. Some barcodes for some genes work with some groups of sponges. However, they're undetectable for other groups. And this is at the class level. So for example, CO1, which is mitochondrial primers that are used for most of the organisms around the reef, will not work for the class Calcarea. And so there's no such thing as a universal sponge barcode There are some regions that are better than others, and it's just a matter of finding more specific sponge barcodes. There's also a problem with the sponge database. So how are you gonna use genomic data with a reference database that doesn't have any data on the sponges that are found for that region? And this is a problem in Hawaii because there's only a handful of sequences that have been deposited for sponges in Hawaii. So it's very difficult to determine what the taxonomy of these sponges in your seawater sample will be. And so what you end up having is a result as presented by this great paper by Nichols and Peter Marco from the University of Hawaii, where you find a lot of sponges on the reef. In the study, they found a lot of reeds from eDNA on the reef, but they were unable to classify the sponges that formed that column that said sponges. We know there's sponges, but what sponges? We don't know, and this is the reason why. So the answer is to develop a well-curated database for sponges in Hawaii, and that's what I've been doing for the last 3 years. We've been collecting sponges, vouchering them with museums, with the Florida Museum of Natural History and the Bishop Museum eventually. And we collected sponges for DNA extraction, for histology. And just to give you a rundown of how the histology part goes, we determine if the sponge has a mineral skeleton. If it doesn't have a mineral skeleton, you have to use more fancy microscopy in order to identify them. If not, if you get lucky and you have a mineral skeleton, you determine the arrangement of these spicules that are these microskeletal structures that arrange themselves in certain ways to give you the skeleton of the ectosome and the quantosome of the sponge. So these are characters that taxonomists have been using for hundreds of years to classify sponges. We take DNA from these sponges and we do phylogenetic inferences based on different genes, and using multi-locus approaches is the best way to attack and to determine the identity of a taxon. We take this information collectively and we compare with other museum vouchers around the world to determine what is the specimen that we have. That's a big problem because specimens around the world are hundreds of years old. They've been preserved in different types of media and different types of fixatives like ethanol. Some are dry specimens, and so in order to get DNA from them is very difficult. But we have to start somewhere, and this is an example of how Hawaii is part of that problem. So this is a timeline of taxonomic assessments of sponges in Hawaii. We have the Laubenfels who collected sponges in the 1950s and fixed samples in ethanol. He had a lot of dry specimens as well, but provided vouchers, which is very important for all the taxons that he collected, which is good if we want to compare them for morphological features. For DNA isolation, not so much. We have Bergquist in 1967 who conducted surveys of sponges, but no voucher specimens were collected, so it's very difficult for us to compare anything. We only rely on whether the sponge was pink, blue, red, yellow, etc., which is very difficult. We have visual surveys that have been done by the state of Hawaii, but no collections. And there has been a recent study that includes morphological data and DNA genetic sequences for only a few records of Hawaii. So we want to try and improve this, and we've been doing this by sampling sponges in Kaneohe Bay for the last 3 years. Kaneohe Bay is a very special place for sponges because it has probably the highest diversity and highest abundance of sponges around the Hawaiian archipelago. The reason being because water in the southern portions of the bay, where the highest density of sponges are found, have high residence times and a lot of river runoff, and have a lot of dissolved organic carbon, which sponges love to feed on. So we've been lucky to collect sponges on 21 reef sites around Kaneohe Bay. We've also collected sponges as part of this long-term monitoring project. It's an experiment that was run by a post-doctoral researcher at HIMB, Chris Jury, and Molly Timmers, who works for NOAA. I've been running this long-term experiment where we have these tanks that are hooked up to a hose that filters seawater from the coral reef that's adjacent to the institute. So we're able to do these very important studies at HIMB by drawing coral reef seawater, feeding through these mesocosm tanks and altering temperature and PCO2 values to resemble ocean acidification and thermal stress for the end of the century for a 2-year period. So we're able to use these ARM units, which are basically settlement plates. We put them in these tanks, and we monitor basically the sponge community of these units in 38 tanks for 2 years for over a 2-month period. And the data that we got was quite fascinating. We collected over 700 samples through this span, and this is what we found. We found over 150 species in Kaneohe Bay, and we're still counting. And so what we have here is a phylogenetic tree on your left, and this phylogenetic tree has been created with 28S sequence data that other scientists can use for eDNA analysis, etc. So these are all vouchered specimens that we have collected, we have provided a genetic resource for, and we're developing the morphological database as we're working with them. What we find is a quite remarkable distinction between the cryptic sponge community and the coral reef community sponges. So we have a high richness of both communities, but very little amounts of OTUs that are shared between these communities, and very, very, very small amounts of OTUs shared between those that have been previously reported. The reason why we haven't found so much overlapping species with those that have been previously reported is because the Laubenfels sampled from very different habitats that were not Kaneohe Bay. There were deep reefs, 130 feet, by dredging, and they just throw a grab sampler, pulled it back, and collected anything they could through those methods. So it doesn't surprise us that there's such little shared OTUs between them. So this is fascinating because through this cryptic habitats, we have found more diversity in an area that's 10 meters squared than 21 sites around the bay. So if you sample this area continuously for 2 years, you're going to find the diversity easily exceeds the diversity of the entire sponges in the bay. Now what's fascinating about this is that there's a lot of potential for high endemism, and these molecular tools have allowed us to actually see what's endemic and what's not endemic. So if we look at the class Homoscleromorpha, for example, 3 species in this class have been identified previously by the Laubenfels. We have found 11 to 15 species of these class Homoscleromorpha, and of particular interest, so in red what you're seeing are those classified by the Laubenfels, the one in green are those that we have found, the ones in black are the reference sequence data that are in the database. And what you can see is that these sponges are very different from one another and different from what's in the database for these different species. So just to give you an example, we haven't seen a phylogenetic tree before. I'm going to just go up here real quick, but these 2 species are the same, and anything that's distant with lines that are horizontal to each other are different species. So SP6 and SP7 are different species, and these are all different species from anything that's in the database. These other species that we have are closely related to things that are in the database. But yeah, that's just to give you an example of the diversity of new species that we're finding. So we're going to focus in on the Oscarella because I think there's something very, very interesting happening here. When we look at the phylogenetic tree, we find these 4 species of Oscarellas that are only closely related to each other. They're not closely related to anything else that's found in the world. So there's very high probability that these sponges are Hawaiian, are true Hawaiian endemics, and not closely related to any other Oscarellas in the world. And this is actually a classic example of a species flock. A species flocks are different species that are confined to a specific habitat, and these are confined to a specific habitat. They're in a cryptic nook and cranny of the reef where you can't move around with a very quick dispersal time. And what happens is that you end up with a lot of species that relate to each other, and there's high endemism in this community. So it's a very interesting phenomenon. As you can see, these are some pictures to show you how difficult it is to classify them based on morphology. There's a lot of them that look the same, and so you need these genetic resources in order to do an accurate classification of them. Now, although we got really excited about this outcome of all these Hawaiian endemics, they're facing really tough challenges with what lies ahead. And so what you're seeing here are observed diversity and abundance of those homoscleromorphs on different acidification and warming conditions. So what I want you to take from this is that this treatment with the gold line that's spread across at 0 is a treatment where CO2 or ocean acidification is high, and that light blue line is the combination of high temperatures and acidification. So you're seeing is that in the entire 2 years when we were sampling these plates, not a single homoscleromorph settled under high PCO2 conditions, and only slight diversity increase and abundance is observed for that treatment that has both high CO2 and high temperature expected for the end of the century. High temperature doesn't seem to impact the sponge community, which is the line in red. I'm sorry, the line in green, and the ambient conditions, which are the line in red, are pretty much at between 1 and 2 species, and the abundance of 3 individuals per plate. So, although we find this fascinating diversity, we're likely to experience a loss in this diversity in the future. It's kind of a bittersweet outcome for this project. But anyway, this is just to show you how the homeless scleromorphs are absent in these high CO2 conditions and are a lot lower in high temperature, high CO2 conditions. So yeah, that's one way in which this project is helping understand the endemics of Caneoheve. Another example that I want to show you is in this order Subaritida. So Subaritida include the Terpiosochinota that I was talking about before. That group of sponges. And so we previously had 7 species, now we have 19 species based on our careful collection over 2 years. And these species consist of macro sponges on the reef and cryptic sponges on the reef, which are really hard to tell apart, as well as the Oscarellas that we saw previously. And some really interesting new functions are being found on these macro sponges on the reef. So these are all new records except for this Hymenaecidon sp1 species, which I think was originally classified by the Laubenfels. It's a sponge that grows with Parietes colonies, but it doesn't overgrow it. It seems to grow symbiotically with it. However, we have this Hymenaecidon sp6, which is very similar in morphology to Hymenaecidon originally described in Caneoheve, that is actually a bioeroder and aggressively overgrows Acropora colonies. It grows Parietes colonies and actually dissolves the calcium carbonate skeleton of Parietes. These are sponges that are new in the bay that no one has ever seen before. And we've also found this phototrophic sponge. Remember the sponges that associate with cyanobacteria that can grow very quick? We have one in the bay that has never been reported before. You can see how it's overtaking this Parietes colony down here in South Bay, South Caneoheve Bay. It's this, all this black is basically the colony being swallowed up by it. So another component that this project has allowed us to see is basically determine which sponges are actually invasive, which sponges are not invasive, right? So Myceligrandis we have always observed as a sponge that's going to take over the reef. It actually kills coral. However, I haven't seen the sponge kill a single Parietes colony, all right? The sponge, okay, the fact that it grows with the coral doesn't mean that it's actually killing the coral, right? So we've observed the sponge, and the numbers on this abundance of this sponge on the left are actually dwindling. And so when we take a close look on it, you can clearly see that the sponge grows within the finger ridges of Parietes, but it never actually overtakes it. However, Monancora clatrata, which is a new introduction as well in the last 5 years, actually overtakes and actually overgrows coral. And the reason why this happens is because they produce these chemical compounds that are actually protecting them from predators in the case of Monancora clatrata. And Myceligrandis lacks these chemical compounds, which gets eaten by predators. So we've done some assays to determine that. What we find is that these chemicals have allelopathic effects with coral, especially in the case of Monancora clatrata on the right. You can see that this is the extract incorporated in a carrageenan gel extract causing bleaching. So what you're seeing here is the difference in time point 1 and time point 2 of the assay, where we take this gel, we put it on the coral, we monitor through PAM the photosynthetic efficiency of the coral through those 48 hours. So what you're seeing is a decrease in the photosynthetic efficiency. The controls are basically at level, but when you expose them just to the control, no extract, actually Parietes bleaches. But when you mix in the Myceligrandis, that bleaching decreases. So Myceli might actually be producing compounds that help corals be more resilient to bleaching. It affects Montipora capitata, and then Monancora clatrata bleaches both Montipora and Parietes. So this project is actually allowing us to see which sponges are actually the invasives and which ones are the intruders. So in summary, I hope you see the importance of establishing this database of sponge biodiversity in Hawaiian reefs. We don't know what the true diversity of sponges are in the reef. We're finding this insane amount of diversity of sponges that are endemic. And when we say that, what are the impacts of climate change going to be? We just found dozens of new species that are potentially going to disappear by the end of the century. This is like discovery of, wow, look at all these species. Oh, we're going to lose them. So we really don't know what's out there. And so establishing a baseline is the correct first step. Correct species identification is also important. We want to manage a resource, manage a sponge that is out there, that is overtaking the reef. You better know what species it is. So in the case of Michaelia grandis and Monochoroca latrata, they're really hard to tell apart on the reef, right? They're both red sponges that grow with coral. So you better know what species you're dealing with if you want to eradicate it. We're also studying the different functional roles, the fact that we're going to lose these Oscarellas. What is that going to mean for the future? They might be very important contributors to that carbon loop that I was talking about at the beginning, providing all these nutrients for the ecosystem. Well, we don't know. So genetic data will be useful for future detections. I plan to publish this paper on over 150 new records for Hawaii using DNA barcodes and voucher specimens so that scientists around the world can use for studies around remote island archipelagos. And we want to continue building that backbone of genetic resources so that we can use this for eDNA, for the most efficient use of eDNA data and metabarcoding analysis. We want to develop an online database with these situ pictures so that state managers can go out on the reef and be like, all right, now I know these 5 different species based on this sponge guide that we have developed. We also want to design specific primers. Like I told you before, the primer issue is an issue for molecular sequencing, so we want to design those primers that are specific for sponge DNA and use them for future eDNA analysis of harbors in Honolulu. So with that, I want to acknowledge Hawaii Invasive Species Council, NOAA, National Science Foundation, Marine Geo for contributing to this project. The Tobo Lab has been incredibly supportive, especially my PI, Robert J. Tunen, who's been a fantastic PI throughout this process. Dr. Gustav Pauly has been handling my 700 sample submissions to the Florida Museum of Natural History. Division of Aquatic Resources has allowed some field time on Honolulu harbors. The Hagedorn Lab has provided some microscopy and some funding for barcoding. The HMB education program has been a part of some of these projects, so I've used some of these topics that I've talked about, used them as a research project for local Hawaiian high school students that are now undergraduates and have exposed them to different experiences in marine sciences by conducting the field assay like you saw, using the 2 invasive sponges, which had different results with, on bleaching on coral in Hawaii. We also attract many international students. Rachidawi Hachai is from Thailand. He came in and worked for a little bit in the lab working on these sponges. We've had some field assistants from other postdocs at HIMB, Angela Richards and Chelsea Council, and have been using facilities in UH School of Medicine and on campus to do this histology work for this project. So with that, you can take any questions you might have. Sorry that was a bit long, but
That's good.
yeah.
Thank you.
Yeah.